Good afternoon, everyone. Welcome to the Wednesday afternoon lecture, which this time is an NIH Director's lecture. So the NIH Director is happy to be here to provide the introduction to a special presenter, Dr. Judith Campisi, who is a remarkable researcher in the area of aging and also cancer, and also happens to be a friend of mine, and I'm really delighted, therefore, to be able to give her this introduction. She is currently professor at the Buck Institute for Research on Aging, as well as senior scientist at the Lawrence Berkeley National Laboratory Life Sciences Division and adjunct professor at USC. She did her undergraduate work and her PhD at the State University of New York in Stony Brook. Following that, did a postdoc at the Dana-Farber with Art Pardee. After spending some 6 years at Boston University School of Medicine as an assistant and then associate professor, she moved to the Lawrence Berkeley National Lab in 1990, where she continues to this day having an appointment, but then in 2002 set up laboratory at the Buck Institute for Research on Aging. Her research has, I think, been remarkably contributory in terms of our understanding of this phenomenon called senescence. One of the reasons that I know Judy is because my own lab works on this rare condition called progeria, which is a dramatic form of premature aging, where cells undergo senescence much too rapidly. And so the children who have those cells likewise have a very shortened lifespan. And the kind of work that Judy has done and her willingness to dedicate herself to helping out with this rare condition by serving in multiple ways with the Progeria Research Foundation is just an indication of how important her efforts have been in shedding light on this common phenomenon. It's happening to some of your cells right now. And boy, we really like to understand that as much as we can. A particular phenomenon, which she will undoubtedly mention, and which I think was a pretty new revelation to many of us, is this whole concept of SASP, S-A-S-P, senescence-associated secretory phenotype, which has taught us that senescent cells are not just sitting there getting in the way, but otherwise not being harmful. They are actually doing some pretty seriously bad things to their healthy neighbors. And the more we can understand that, the better shot we will have in getting a full grasp on how we might give more opportunities to more people to experience healthy aging. So it is a remarkable opportunity to bring special presenters to NIH on Wednesdays. Today is a nice example of that, to put it mildly. Please join me in welcoming Dr. Judith Campisi.
So thank you, Francis, for this very kind introduction. Actually, I'm delighted to be here. I have lots of friends here and colleagues and hopefully more after today. So this is just a classic disclosure. I'm not going to be talking about anything the company is doing, but I will start with aging. And so let's see, how many people in the audience would like to live a long life? Good. You know, this is what you have to look forward to. So this is obviously a partial list of the very, very many diseases that are considered age-related diseases. And what is so remarkable about these diseases, so very different tissues, very different manifestations of what goes wrong, and yet all of these diseases have one thing in common. They're extremely rare in young people, and somewhere around the midpoint of the lifespan, so 50 to 60 years for people, all of these diseases rise with exponential kinetics. Now, those of us who work on aging, we think it's not a coincidence. And we think it's because there are probably basic aging processes that are driving all of these diseases and can account for many of the phenotypes we associate with aging. So as Francis pointed out, today I'm going to tell you a story of one such process that we think qualifies for being a basic aging process. It's called cellular senescence. I'll explain a little bit more what it's about. I don't want to leave you with the idea that we think this is the only thing that drives aging, but I think we can now say that this is probably one of the things that is driving multiple manifestations of aging. So what is cellular senescence? I'm going to show you two ways that people in the field think about senescence, and then I'm going to show you a third way, which I think is actually the more accurate way. So what happens when cells become senescent? So this is a cell fate, and this cell fate can be adopted by cells in response to a variety of stresses. So what's important about these stresses is they're common drivers of two things that we know about physiologically. One is cancer, and the other is aging. And the fate that the cells adopt is to undergo what we call a tripartite phenotype. So the cells arrest growth, they stop dividing, and this is good, because this is why these stressed or damaged cells will never go on to form a tumor, because they can't divide again. But coupled to this growth arrest are two other phenotypes. The cells tend to resist apoptosis. They can die. I'll show you that we can get them to die. But in general, they prefer not to die, so they're fairly stable. And then they begin to secrete a very wide variety of molecules, which I'll tell you a little bit more about as the lecture goes on. And so this tripartite phenotype then can be induced by lots and lots of things that are considered stressors. And of course, many of these stressors occur during aging. But there's a second way to think about senescence, and it's kind of important because it gets at the evolutionary origins of why this phenotype evolved. So there are now two labs that have shown that there are waves of senescence in embryos, both mice and humans. And at least in mice, it can be shown that those senescent cells are responsible for fine-tuning the morphogenesis of certain structures in the embryo. Ramanan in Texas has shown that labor begins with a wave of senescence, and women who undergo early labor have an early wave of senescence, and women who undergo late labor have a late wave of senescence. So it seems to be important for initiating labor. And then finally, we and other labs have shown that the secretory phenotype in particular is very important for optimizing tissue repair and wound healing. So really, I think the way we need to think about senescence is that it's an evolutionary trade-off. There are positive aspects to being able to undergo this cell fate, and we think that this was the evolutionary pressure to develop this cell fate. But then I'll show you late in life, the same processes that are healthy for us when we're young can be deleterious and drive aging phenotypes and a large number of age-related pathologies, at least in a mouse. And so this is the downside. I should also point out that this idea that there can be something that was under evolutionary pressure that's good for you when you're young, but that same thing can be bad for you when you're old, is many decades old. It was first described by George Williams, and he termed this process antagonistic pleiotropy. And so I'm going to argue that senescence is an example of antagonistic pleiotropy. It also makes trying to undo the bad things that senescent cells do somewhat challenging. So let me very briefly say something about how we can identify senescent cells. There are now literally more than a dozen different markers that many labs have contributed to. So we can, some of them are listed here, some of them involve the molecules that are secreted, but there are things that happen within the cells that allow us to identify senescent cells. I do want to point out, though, that so far as I can tell, there is no such thing as a senescence-specific marker. All the markers have lives outside of senescence. And so this is a characteristic of the senescent state. Those of you who are thinking about working in this field, take heart if you just have to look at multiple markers in order to have some confidence that a cell is indeed senescent. But using these markers, a number of labs now have asked the question, when and where do you find these cells that have adopted this senescent cell fate? And the answer is twofold. The first is they increase with age. So far as I know, this has been shown in virtually every vertebrate species that has been examined, that senescent cells increase with age, and they increase in multiple tissues. Actually, also every tissue that has been examined has been shown to accumulate senescent cells with age. This is human skin. This is one of these markers. It's an enzyme, and this is basically a beta-galactosidase. So this is the blue staining of a dye that the enzyme converts into an insoluble precipitate. And you can see it's present in the dermis and the epidermis of older skin, but very rare in young skin. In this experiment, young was under 30 and old was over 50. So I probably just got half the audience here depressed, but that's okay. I'm going to cheer you up at the end, I hope. Okay. But the other place that number of labs have now shown that these cells are present is at sites of age-related pathology. So if you compare diseased tissue with age-matched healthy tissue, there tend to be more senescent cells in the diseased tissue compared to the age-matched normal tissue. So here, for example, is these are smooth muscle cells from the pulmonary artery of patients with chronic obstructive pulmonary disorder, the major cause of lung failure in the elderly. And this is age-matched control, so there's always seems to be more in the diseased tissue. This is an interesting example. This is autopsy material, obviously, from a patient with Alzheimer's disease. And if you compare the brains of patients with Alzheimer's disease versus an age-matched brain that is not diseased, there seems to be more senescent cells in the diseased brain. But what's interesting, for the most part, what are the cells that are senescent are the astrocytes, not the neurons and not the microglia, but the astrocytes. And of course, I probably don't need to remind you that astrocytes are one of the cell types that can give rise to brain cancer. So again, suggesting and enforcing the idea that this is an anti-cancer mechanism. So in thinking about this evolutionary balancing act, I'm going to first describe very briefly what is known about this growth arrest, and then tell you more about this secretory phenotype, because we think this is what's important for driving aging and age-related diseases. So the growth arrest has been, that was first studied actually by a number of major labs that were working on the cell cycle, and it's clear now that this growth arrest is imposed by what are arguably the two most powerful tumor suppressor pathways that we have encoded in the human genome, or the mammalian genome. And that is the pathway that's driven by p53 and the pathway that's driven by the retinoblastoma protein. So both of these proteins are crucial tumor suppressor proteins. They control the cell cycle. And most importantly, you'll see, I'll come to this point in a minute, is this protein called p16-INK4A. It is a cyclin-dependent kinase inhibitor, and it is important for keeping the retinoblastoma protein in its tumor suppressive form, its unphosphorylated form. These pathways can independently drive a cell into senescence, but they also tend to talk to each other. And so often you see both, for example, p21 and p16 in senescent cells, but they can also sometimes act independently. The secretory phenotype is actually even more complicated than those pathways. So I'm going to talk you through what we've learned about this secretory phenotype. We first discovered it using antibody arrays. It was more than 10 years ago, and there were no high throughput ways of looking at secreted proteins. And we defined it as containing about 40 different secreted proteins. So now with unbiased mass spec analysis, we've been able to expand that by more than tenfold. So the cells are secreting more than 400 different molecules, both proteins as well as small molecules, small metabolites. So what we know about the secretory phenotype is that it is very energy demanding and energy consuming. So you may read sometimes in the lay press, they refer to senescent cells as zombie cells. they're not zombies, they're ballerinas, and they're expending a huge amount of energy because they're secreting all of these molecules. It's highly complex. As I said, now there are more than 400 proteins that have been identified. Some of them are in exosomes. Also, the phenotype is extremely heterogeneous. I'll show you an example about that. And we now know from a number of cell culture experiments and mouse experiments that it is the secretory phenotype that is most important for driving aging pathologies and age-related phenotypes. So this phenotype, it's a so-called senescence-associated secretory phenotype. It will vary with cell type. I'll show you an example of that. It's dynamic. It changes over time. And it depends upon how the cells were induced to become senescent, whether it was something through metabolism or DNA damage or any of the other inducers. So let me first show you an example of the cell type specificity. This is an antibody array. And so everything in yellow is highly secreted, and everything in blue or black is either no change or is secreted at low levels. And this is an experiment that was first done by Jean-Philippe Chappé. And what he did was he took fibroblasts and epithelial cells from the same tissue. In this case, it was a prostate, a human prostate. So the genotype is the same, but the cell types are different. And he induced the cells to become senescent using ionizing radiation. And so what you can see is there are things in common that are secreted by both types of cells, both the stroma and the epithelium, but there are things that are stromal specific and there are things that are epithelial specific. And we've seen this now looking at dozens of different human and mouse cell types, that each cell type seems to have its own complement of molecules that it secretes when it becomes senescent. It's also dynamic. I'm showing you an example now of two genes that are induced upon an ionizing radiation stimulus to induce senescence in human fibroblasts. One of these genes codes for a leukotriene, and I'm going to come back to this point in a moment, but just to remind you that leukotrienes, this particular leukotriene is pro-fibrotic, and you do need an initial fibrotic response in order to heal a wound. But you can see what happens. There's a very early increase in the expression of this gene, and then it falls. It slowly comes back again, and eventually it comes down to baseline. This is the expression of a gene that codes for a prostaglandin, which is anti-fibrotic. It's something that helps resolve fibrosis. And you can see that there's a long lag period, and then slowly the expression of this gene rises, and it stays high as long as the cells are senescent. And we've seen lots of examples now of genes that seem to change over time with different dynamics depending upon the physiological context. And then finally, it really depends on the inducer. So, whoops, let's see. There we go. It really depends upon the inducer. What I've listed here for you are 4 different ways that we can drive cells into a senescent state. Genotoxic stress. Many oncogenes will drive cells into senescence. This is consistent with the growth arrest being tumor suppressive. Just overexpressing those cell cycle inhibitors like P16 or P21 will cause a senescence response. And mitochondrial dysfunction also will drive cells into senescence. And what I've done here is just code for you different patterns of secreted molecules that are dependent upon how the cells were induced to senesce. Actually, right now, we don't know in vivo, certainly not in humans, how senescent cells are driven into the senescent state. And we're sort of now trying to get at that question by characterizing the secretome that may give us hints as to whether it's mostly mitochondrial dysfunction or mostly DNA damage. And so we'll see those experiments are ongoing. And the last point I want to make about the senescent state is it is surprisingly heterogeneous. So we've done the following experiment. We've taken normal human lung fibroblasts and treated them with a chemotherapeutic agent, doxorubicin. This is a DNA damaging agent, and it's known to cause, actually, I just left a workshop that was a collaboration between the NIA and NCI on trying to understand why certain chemotherapeutics seem to accelerate aging in a patient population. So this is one of those chemotherapeutic agents. It damages DNA, it causes cells to become senescent. So same genotype, same environment. This is all done in tissue culture. And now what we've done is we've done single cell analysis of these two populations. So I don't know how many of you are used to dealing with single cell analyses, but basically what happens is you can ask the computer to cluster cells based on their pattern of gene expression and then code them in different colors depending upon how similar they are. So these are non-senescent cells and these are senescent cells, and as you can see, they're well separated, and that's what you'd expect. We know that these cells have different patterns of gene expression, so we would expect them to be separated. So this is at low resolution. If you now ask the computer to dwell into those transcriptomes at higher resolution and ask whether there are subpopulations, you get a pattern that looks like this. So these are different subpopulations. So these are the senescent cells, and you can see that they're segregating from each other, and there are even some that are really very well separated from the others. So we're now trying to understand what these patterns mean, and we're hopeful, and this is only hope, we have no data yet, that maybe we can say some of these subpopulations are the good ones that are promoting wound healing and tissue repair, and others are particularly bad. This will make it easier to target drugs to try to eliminate the bad ones and keep the good ones. But this is still a dream. We're really not there. So everything I've told you so far suggests that senescent cells are sort of a smoking gun. They're present at the right time and the right place to qualify for being one of these basic aging processes that's driving these different diseases. Actually, I used to have a slide of a smoking gun, and I've just decided this is the wrong time to be showing a smoking gun. This is a real winery, though, in Napa Valley. So those of you, I haven't tasted the wine, so I don't know if it's any good, but there is a real winery with this name. So the two questions now that we next had to try to address is, what's the how is it possible that these cells could be driving such disparate diseases? And then, of course, do they actually do anything? Because everything I've told you so far is correlation. So let me start with how, and I'll tell you what our overarching hypothesis for the mechanism by which a senescent cell could be driving very disparate pathology. And that has to do with the secretome. We tend to bin the secretome into two different groups because they're under different molecular control, but what both of these groups have in common is they have the ability to drive inflammation. And so every one of those diseases I showed you on my first slide has either as a cause or an exacerbator chronic inflammation as a very important component. And several years ago, Claudio Franceschi coined this term, inflammaging, to characterize what happens to tissues when they become aged. And most pathologists will tell you that you can find in old tissues, but not in young tissues, evidence of chronic inflammation as judged by low-level infiltration of immune cells. And I probably also should point out that it's well known that inflammation, chronic inflammation, can destroy tissues because of the bioactive molecules that innate immune cells are making, but it also can disrupt normal tissues from functioning. It can prevent stem cells from functioning. And of course, it is a major risk factor for the development of cancer. That's the bad side, but the good side is you also do need inflammation in order to initiate wound healing and tissue repair. So again, it's this tension between the good and the old. So I will show you now a little bit more about the secretory phenotype and then some of the evidence that it actually does something. So the first arm of the secretory phenotype is the so-called p53 dependent arm. And what is striking about this arm is that it is totally independent of changes in transcription. We would never have picked this up with any of our high-throughput transcriptome analyses. And it was first discovered by Albert Davalos. He was studying a major nuclear protein called HMGB1. This is a protein that binds DNA sequence independently, helps bend DNA, and helps transcription factors access their binding site. In a separate world, immunologists had been studying HMGB1 because macrophages secrete this protein when they're about to initiate an inflammatory event. And what Albert found is every senescent cell he's examined loses the protein from the nucleus and secretes it outside the cell. So HMGB1 was the founding member of a family of proteins that were initially called alarmins, but now they're called damage-associated molecular patterns, or DAMPs. And we now know that this was the tip of the iceberg. There are now a number of DAMPs. I'm just listing a few here that senescent cells secrete. And again, the primary function of DAMP secretion is to alert the tissue to danger and initiate an inflammatory response. The second arm of the secretory phenotype is, again, this was discovered by Jean-Philippe. Again, it's an antibody array, and what you're looking at are 5 different human fibroblasts from different tissues, breast, lung, and skin, and they're all made senescent by ionizing radiation, but actually it doesn't matter how we make them senescent, we get this kind of pattern, meaning lots of things that are secreted by the senescent cells compared to their non-senescent counterpart, but also lots of plasticity. So it depends on both genotype, tissue of origin, and also, as we found out later, timing after initiation of senescence. This part is mostly due to the transcriptional upregulation of the genes that encode these proteins, and it's completely independent of the p53 tumor suppressor. So this, I think, is a good clue as to how senescent cells might drive aging by virtue of this inflammatory response. Of course, the real question now is, do they actually do anything? So first I'll show you 3 examples in culture, and then I'll show you a mouse model that gives us confidence that the answer to this is yes, because Francis would have told me to stay in Berkeley if the answer was no, I think. Okay, so first example. This is a 3-dimensional culture of mammary epithelial cells from a mid-pregnant mouse. We seed them as single cells. They undergo limited cell division. They form these beautiful alveoli. We add lactogenic hormones to the culture, and the cells produce milk. So the red staining is beta-casein. If we embed in these cultures non-senescent stromal cells, we still get these beautiful alveoli. But you can see what happens if we embed senescent stromal cells. These alveoli are misshapen, milk production has failed. So this argues that something the cells are making are disrupting normal tissue structures and functions. This work was done by Simona Paranello, and she was even able to identify the molecules that were primarily responsible for this decline. And it's mostly the matrix metalloproteinases that the cells are producing. The second example is what happens to stem cells. These are human neural stem cells, or myoblasts. And what we've done is taken conditioned medium from either astrocytes for the neural stem cells or fibroadipogenic progenitor cells in the case of the muscle myoblasts, and now asked whether they can differentiate. So in the case of the muscle, we get lots of differentiation, but if that conditioned medium is senescent, we really block a large part of their ability to differentiate. In the case of the neural stem cells, this is cell proliferation. This is BRDO incorporation. Something the cells are making, these are senescent astrocytes that we've collected the conditioned medium from, and there's something those astrocytes are making that suppresses the ability of those neural stem cells to proliferate. We don't know what these factors are. We're still working on that. And the last example I'll show you is something that was first discovered by Anna Krtolika many years ago, and it really shocked us. We didn't quite expect to find this. This is tumor volume over time of a pre-malignant breast epithelial cell line. We inject it into the mammary gland of the mouse. They're pre-malignant. They don't form tumors. But in the presence of senescent mammary fibroblasts, these cells will convert to full-blown malignancy. This is an immune-competent mouse. If we go to an immune-incompetent mouse and inject human breast cancer cells into the mammary gland, this is what the tumor looks like after 6 weeks. This is what it looks like if we co-inject with senescent stromal cells. These cells are also much better vascularized than these cells, and they metastasize because senescent cells make a lot of VEGF. So the model then is, with age, we're accumulating senescent cells, and we would argue that these cells, by virtue of what they secrete, will affect neighboring cells and cause those cells to fail to function. And that would explain some of the degenerative diseases of aging. And of course, what really happens is we're all accumulating premalignant mutations. So we would argue then that senescent cells could also, by virtue of their secretions, fuel the conversion of premalignant cells into tumor cells and therefore explain late-life cancer. Just to remind you, cancer is as much an age-related disease as many of the other degenerative diseases. So if you don't have senescence, you will die of cancer. And if you do have senescence, you're still going to die of cancer. So what do we do about this? So we actually thought of 2 strategies for how we might intervene in this process and improve at least health. So the first was, what if we can just get the cells to stop secreting? And I can tell you right now, we've pretty much abandoned that approach as an approach, although we're still studying it from the point of view of basic biology. And the reason is, we did what every good cell and molecular biologist did. We drilled down, we identified the pathways that drive these secretions. Those pathways were known. They had target genes and therefore target proteins. And what was great is there were already drugs that could target some of the players in these major pathways. So here are the 3 pathways that drive the secretory phenotype. The DNA damage response pathway, this NF-kappa B P38 MAP kinase pathway, and of course the mTOR pathway, which is important as a longevity pathway. The problem is we can inhibit these pathways, but this is not something you would want to do in a healthy organism because all of these pathways are important for maintaining normal tissue homeostasis. The other problem with this approach is, as I said, there are drugs. So we got the drugs and we put them on the cells stopped secreting. It was great. We did this both in vivo and in culture. And then you take away the drug and the cells start secreting again. So drug companies love this because it means you have to take the drug all the time. But we felt that this is not a way to approach aging. I would argue there's no such thing as a perfectly safe drug. And so it really didn't seem practical to be able to use these drugs to try to improve an aging phenotype. So yeah, so that's the other problem. So then we thought, let's take a different approach and let's try to get the cells to die. Let's try to overcome this resistance to apoptosis and get them to just go away. And then you could have them slowly accumulate and maybe hit them only intermittently and let them die. So we made a transgenic mouse model that enables us to do this. So what we did, so this is a transgene. This is not a knock-in. So the animals have normal copies of every gene, but they have this transgene. It's a bacterial artificial chromosome that contains that P16 INC4A locus. And we simply had the promoter for P16 driving this fusion protein, which is totally artificial, but it contains a luciferase, so we can image senescent cells in living animals. It contains red fluorescent protein, so we can sort the cells from tissues. But most importantly, it contains the herpes simplex virus thymidine kinase, which will convert the prodrug, ganciclovir, into a phosphorylated derivative that is a potent DNA chain terminator. Now, senescent cells don't replicate nuclear DNA. They do replicate mitochondrial DNA. And Remy Laberge was able to show that this derivative gets into the mitochondria, fragments the mitochondrial genome, the cells die by apoptosis. So Marco Di Maria did most of this work in characterizing this mouse model. I'm just going to show you one experiment. This is whole body luminescence in a mouse. So at 12 months of age, so that's somewhere between 30 and 40 years for the, in human terms, the signal is very low. And then shortly thereafter, we get this essentially exponential rise in luminescence. And we can eliminate about 70 to 80% of that signal by treating the animals with ganciclovir. And this is just visceral fat, showing you again that beta-galactosidase. Visceral fat has a lot of senescent cells. If we treat the animals with ganciclovir, again, about 70 to 80% clearance of the cells. So there are two mouse models that can have this capability. One was made by us, and the other was made by Jim Kirkland and Jan van Dersen at the Mayo Clinic. Their transgene is a little bit different from ours, but the same principle, P16 driving a killer gene. And between our groups, we have now shared our mice with literally dozens of labs, each of which is focused on a different age-related disease. And at this point, this slide is already out of date. I just can't keep up with all of the reports now showing that at least in a mouse, senescent cells are causal for certain age-related pathologies. So I've listed them here, everything from Alzheimer's and Parkinson's disease to osteoarthritis, osteoporosis, et cetera, et cetera. I'll show you also that, again, the cells are important for wound healing. We have used the mouse to demonstrate that. But this is, again, a wide range of age-related pathologies, and it really, I think, tells us that we can have some confidence that cellular senescence is a driver, a major driver of basic aging. It is a basic aging process that drives a large number of age-related pathologies, at least in a mouse. So I'll show you just a little bit of data from our lab. We're very interested in the premature aging that has been reported and is being reported more and more now as a consequence of genotoxic chemotherapies. This is also true actually now for certain drugs that are used to treat HIV/AIDS. And so what we did was, this again is Marco Di Maria's work. He gave our mice a single dose of several genotoxic chemotherapies and cytotoxic chemotherapies, and then imaged the animals. So initially there's a big burst of senescent cells, and then it begins to decline, but it never really goes away. These animals seem to have a long-term burden of senescent cells in their body. And now we can ask, what is the consequence of having that burden? And the first has to do with cancer metastasis. So what Marco did was he took a breast cancer cell line labeled now with a different luciferase. So now we're going to follow the tumor in vivo with firefly luciferase. It's known that if you take these cells and inject them into the inguinal mammary gland of the mouse, they metastasize to the lung and the liver. So the experiment is we give the animals breast cancer, then we give them chemotherapy, in this case again, doxorubicin, and a few days later we either clear senescent cells or not and ask what happens to the tumor. So here's a control animal, here's the primary tumor, here's a metastasis to the liver and the lung. Here's what happens if we clear senescent cells after the chemotherapy. So we have variable effects on the primary tumor. We actually don't really understand this. But in all cases, we completely prevented the ability of those cells to metastasize. Our hypothesis is that it's because the secretory phenotype is changing the milieu of distal tissues and therefore making those tissues incapable of supporting metastatic growth. But it's also possible that we're changing something about the primary tumor. We still haven't sorted that out. The other problem with chemotherapy, especially the anthracycline drugs like doxorubicin, is cardiac dysfunction. We can measure that in a mouse by echocardiography. So what happens is doxorubicin will cause a decline in the ejection fraction and fractional shortening. But if after the chemotherapy we give them ganciclovir, we can prevent this decline in cardiotoxicity. Another problem is blood clotting, throwing off blood clots as a consequence of the chemotherapy. We can measure this in a mouse. We just nick the tail and measure the tail bleeding time and the hemoglobin in PBS. So doxorubicin will shorten the bleeding time, so it's pro-clotting. And again, we can prevent that if we eliminate senescent cells. So we're cautiously optimistic that this might be something the oncologists might want to think about to eliminate some of the pro-aging side effects of certain chemotherapeutic regimens. Now, I'm just going to show you one other example of a very different disease, which is Parkinson's disease. This was a collaboration with Julie Anderson, who is a neurobiologist at the Buck Institute. Julie was studying autopsy material from humans, substantia nigra from patients with Parkinson's disease or age-matched controls. And what she noticed is that the diseased tissues seem to have a higher level of a number of senescence markers. And so we thought we should try to test the idea that senescence might be driving certain aspects of Parkinson's disease. So it turns out you can do this in a mouse by treating the animals with paraquat. This is an herbicide. It causes oxidative stress, and actually it's banned now in California, but it causes Parkinsonian symptoms in both mice and humans. And when we added paraquat to either cells in culture or animals, what we notice is that paraquat induced senescence primarily of astrocytes in the brain. And so now what we could do is give the animals paraquat and then clear senescent cells or not and ask what happens to motor neuron function. So here's a rearing assay that the mouse must do in order to, it requires some motor neuron coordination in order to do this rearing. After paraquat, the animals can't do it. But if we clear senescent cells shortly after we administer the paraquat, we can now prevent this loss of motor neuron function. So again, I want to emphasize that I think we're gaining some confidence that cellular senescence might qualify for being a basic aging process that can drive very different types of age-related diseases. So the last thing I want to talk about, it gets back to the evolutionary question. Why on earth did this phenotype evolve? If you think about it, apoptosis is a really good tumor suppressive mechanism because you have cells that die, they can't go on to form a tumor. So why did we need this messy, messy phenotype, especially with the secretions that these cells are producing? And the answer is, especially those secretions are important for wound healing. So this is an example of luminescence at the site of a 6 millimeter punch biopsy that we do on the dorsal flank of the animals. And you can see that there's a peak of senescence at about 5 days after we wound the animals. These are female mice. I'm going to show you data in male mice. Males and females don't heal their wounds with the same trajectory, so males are slower than females, so this is female mice. And we can prevent this peak of senescence by treating the animals with ganciclovir. And now we can ask what happens to wound healing. And it turns out that wound healing is retarded. So here are wild-type animals. They're healing their wounds in 5 to 6 days. Here are the senescent-free wounds, and clearly they're taking longer to heal, and also these wounds are more fibrotic than these wounds. So now, because we can sort cells from our transgenic mice, Marco de Maria sorted the cells from the wound and tried to figure out what senescent cells were making that could be so important for wound healing. And he was fairly lucky in that he identified a single gene that was turned on, which codes for a relatively understudied isoform of PDGF. This is PDGFA. And you can see now, so these are male mice now. So here are male mice healing their wounds. Here are the senescent-free wounds. They're slower, but now he just topically applies PDGFA to those wounds, and he can completely reverse that slow wound healing phenotype. So one thing is puzzling about these data. I showed you that senescent cells accumulate with age in human skin. Everyone knows wound healing doesn't get better with age. So how do we reconcile what I've just shown you with what we know happens in the clinic? To do this, we collaborated with Simon Meloff, who's also at the Buck Institute. He had a transgenic mouse model in which he could conditionally remove the superoxide gene that codes for the mitochondrially localized superoxide dismutase. And it's under a skin-specific promoter that's activated by tamoxifen. So when we excise SOD2 from this mouse in the skin, we get senescent cells in the skin. But these cells now are chronically present. And now we can ask what happens to wound healing. And it turns out wound healing is again retarded. So here are the animals with chronic senescent cells versus those that don't have the presence of that senescent cell. So the idea then is that what evolution selected for was the transient presence of these cells, and that what happens with age is they become chronically present, and that is what could be driving the pathologies that we see. So I want to end with a question I always get. Okay, we're not going to make transgenic people, at least not soon. Are there drugs that can do what our transgene can do that hold hope for people? And the answer is there are. There are a number of labs and a number of companies now that have identified a class of drugs. They're called senolytic drugs, and they tend to target proteins that allow senescent cells to become somewhat resistant to apoptosis and force them to die. And so the first of these drugs has just completed phase 1 clinical trials, and so I think this is reason for some hope for the future. The question is, will they actually impact our lifespan? They certainly have good promise for impacting our health span, but what about lifespan? So that experiment has been done in the mouse by Jan van Dersen, and this is from his publication. These are his transgenic mice. These have senescent cells. These have no senescent cells. He eliminated senescent cells regularly throughout the lifespan of these mice. And he got a very impressive increase in median lifespan. So the animals died healthier, but there was no effect on maximum lifespan. So everyone asks him, what do these animals die of? And it turns out they die of a range of pathologies. It's not one thing that's killing these animals. So I'll just leave you with some thoughts about the promise of health span and the possibility that we will have an impact on lifespan, which I think is much, much less. So think about C. elegans. 1,000 cells, very simple organism. The world record for extending its lifespan is 10-fold. And then you move up the evolutionary scale a little bit to Drosophila, and there, less than 2-fold increase in lifespan. Any manipulation, whether it's genetic or pharmacological. And then you move to the mouse. I even doubt this 40%, but certainly lifespan extensions of maybe somewhere between 10 and 30%. Actually, if you get a 20% increase in a mouse lifespan, you have a paper in Nature, Cell, and Science all at once. So I'm not sure. I think evolution is telling us something about how Species-specific maximum lifespan is set, and it's probably set by modifying multiple genes, and we may not soon have a drug that can target all of those genes. So I'll end with this idea. So here is Jeanne Calment. She's the woman that was presumably the oldest documented human that walked the face of the earth. This is her on her 121st birthday. I don't think many of you would want to age the way she aged. She had cataracts, she had sarcopenia, she had osteoporosis. I think what we want is to emulate this guy. Does anybody recognize him? This is Thurgood Marshall, the first Black Supreme Court justice in the United States. It's a lifetime appointment. Somebody had the nerve to ask him how long he plans to live. This was in the 1960s. And this is his reply, and I think this might be possible. So I will stop here and thank the many people in the lab, past lab members, and an incredible group of collaborators without whom none of this would be possible.
Terrific, Judy. Please, we have time for questions, and use the microphones in the aisles as usual so people listening by video can hear. We can start right over here.
Yeah, a long time ago in pathology, we used to look at lipofuscin, pigment in the heart, and we used to say, this is what's the big deal. And they would see it in the brain, and you'd talk it in Purkinje fibers and all over the place and say, if you can eliminate this, you can eliminate the problem of senescence and aging, and it was actually considered a space-occupying lesion.
Yes.
Now, why aren't you simply just saying, hey, we got lipofuscin, we're going to figure out a way to dissolve it, and problem solved?
Because you can do it. You can do it by regulating lysosomal behavior. And what that will do is that will make the cells look better, but it won't stop them from secreting.
The other, since nobody else, there is somebody else, then I'll just say thank you.
Yes, please.
Towards the end of the transient wound healing process that you showed, what happens to the senescent cells that were recruited? Do they die, or do they become non-senescent, or something else?
So it's a great question, and it has two parts. One part is in the skin. What happens is some of those cells actually get trapped in the scab, so we can see the scab is luminescent. So it's probably mechanical loss. But Scott Lowe has looked at liver injury where there's no scab, and he's been able to show that natural killer cells will identify at least a subfraction of senescent cells and will actively kill them, because senescent cells tend to express ligands for the activating receptors on natural killer cells. So that's also part of the mechanism. The question then is, why do they accumulate with age? We don't lose natural killer cells with age. And so this is an open question in the field, and we're still trying to figure that out.
A related question to that, Judy, is what about embryonic development, where clearly this whole process must be important for modeling tissue? What happens to those senescent cells as tissues do get developed and reshaped? Do they stay there, or do they also get wiped out, and if so, how?
They also get wiped out, but the mechanism is not known. So we know that they're transiently present in the embryo, and then at some point you don't see them anymore.
Yes, please.
So there's a heterogeneity between in the secretary profiles. Are there any known systematic differences between the profiles for pro-damage senescent cells and those senescent cells involved in tissue repair?
Yes, so it's a great question. The short answer is we don't know yet. So we're actually doing those analyses now, trying to see if there's a profile that says I'm pro-regeneration and I'm pro-inflammatory or pathological inflammatory. And we just don't have the answers yet.
It seems a little intriguing that these cells would suddenly begin to overexpress 400 plus proteins that all get secreted. Is there something that brings these together? Is there some mechanism that's bringing together the fact that they're just being secreted, or are these cells also expressing things that get misfolded, accumulated in the cell, things other than secretion for these hundreds of proteins.
So autophagy is upregulated, probably because there's great danger of getting this kind of protein aggregation. And if you inhibit autophagy, the cells are in deep trouble. They'll die. And inhibiting autophagy is also not a good clinical strategy. As you know, this is a highly balanced process. But yeah, they're under ER stress and Golgi stress because they're secreting so much. And they have to upregulate autophagy in order to handle that. And they do.
Great talk. Would you say the senescent cells are the dominant driver of aging rather than telomeres?
So telomeres drive cells, short telomeres drive cells into senescence. So I think the more accurate way to think about telomeres is when a telomere fails, it's a form of DNA damage. And DNA damage, we know, drives cells into senescence. So, and I really don't want to give you the impression that we think senescence is the dominant form or the only form of driving aging. As you saw, you can eliminate senescent cells throughout the life of a mouse. They don't really live longer. They just die healthier. So there are probably other processes that collaborate with the accumulation of these cells.
Well, Judy, this has been a wonderful presentation. We will have a reception in the library for people who wish to continue the conversation about how we can all die healthier. So please join me again in thanking our speaker.
Thank you.